Hi, I'm Jill Venton. I'm a professor in the Department of Chemistry and also associated with the UVA Brain Institute and the Neuroscience Graduate Program. The research in my lab centers around analytical neurochemistry. We develop small electrical chemical sensors, such as this microelectrode made out of carbon nanotube yarn, and even nanoelectrodes like this one made from a carbon nanopipette. And then we apply them to real-time measurements of neurotransmitters in the brains of model organisms, such as small rodents or fruit flies. To highlight some of our recent work, we've recently published a paper where we 3D printed carbon electrodes for the first time. To do this, we use a printer that relies on nanolithography that can precisely print the structures, and then we pyrolyze them to make them carbon, which is a good material for detecting neurotransmitters. As you can see by these SEM pictures of our electrodes, we can make all sorts of shapes like a sphere and a cone, and we can even put little surface features on the edge like these 1 micron diameter nano pillars, and so this is going to allow us to really customize the surface. We use these electrodes then for our in vivo measurements of things like dopamine, and dopamine is an important neurotransmitter in diseases like Parkinson's disease and addiction. To apply our research to real model organisms, we've become really well known in our lab for being able to make neurotransmitter measurements in the fruit fly. This is the life cycle of a fruit fly here, and most of our measurements we've made in this particular stage, the third instar larva. From our collaborator Barry Condren, we learned how to stain this third instar larva so we can see the individual neurons in the brain. But we were the first people ever to be able to make any neurotransmitter measurements in the fruit fly brain. I sometimes like to joke that I don't think anybody else was trying, but in fact it's actually a very important problem. The biologists love to use fruit flies as a genetic model of disease. And so now that we've developed a method that we can insert our tiny electrode into the brain and then measure neurotransmitters such as dopamine, we'll be able to take these measurements now and apply them to disease models, again, such as Parkinson's disease that biologists have already made in the fly. Of course, no research is really ever done by just one person, and so I have a great team of people who help with this research, from graduate students to undergraduate students to postdocs and research scientists.